Welcome to Excellent Grades Academy. This is Dr. Byson EM. Welcome to a world of physiology. Today we're looking at synaptic transmission across a neuromuscular junction. It's an important topic in physiology. Okay, so let's look at this. So, in order for you to understand the synaptic transmission across a neuromuscular junction, you must understand what a neuromuscular junction is. But before we look at that, let's revise some nervous system. So the nervous system is divided into two things. Okay. So it's divided into the central nervous system, which is known as the CNS. Central nervous system. Okay, and the peripheral nervous system. The central nervous system is made up of two things. Okay, it's made up of the spinal cord, and the brain. So outside the brain and the spinal cord is where you find the peripheral nervous system. The peripheral nervous system is made up of the somatic nervous system, and the autonomic nervous system. So autonomic nervous system. The autonomic nervous system is made up of the sympathetic nervous system and the parasympathetic nervous system. Sympathetic. So you have to know the breakdown of a nervous system like this. All right, because they have a different set of cells that are found there and different sets of neurons that are found there. Okay, now let's look at the types of neurons that we find in the nervous system. Okay, so types of neurons. The classifications I'll give here are about polarity. Okay, so number one, you found multipolar neurons. So you find multipolar neurons. Multipolar neurons have got multiple processes. So multipolar neurons. And then you find also bipolar neurons. So bipolar neurons, and you can also find unipolar neurons. So if I was to draw a multipolar neuron here, it would be like this. If this is a cell body, this cell body will have multiple processes emanating from it, coming from it. So this will be a process, this will be a process, this will be a process, this will be a process. So these processes here are dendrites. This is a nucleus here, and then that is the cytoplasm there. Okay, cytoplasm. And then you've got the axon here. This axon would terminate in what we call the axonal terminals. So those are axonal terminals. So these axonal terminals are the structures that will form synapses with a muscle cell. Okay, so this is a multipolar neuron. Why? Because it has multiple processes that are coming from it. So these processes here are what we call dendrites. So these are dendrites. And then this is the axon. So the axon can have Schwann cells in the peripheral nervous system that are surrounding it to provide insulation in form of myelin sheath. Okay. And then these are nodes of Lanvier here. So this is a rough drawing of how a multipolar neuron looks like. So this is a multipolar neuron. I'm not a very good artist. This is the best that I can do. Bipolar neurons just have two processes that are coming from the central nervous system. Okay, just two processes that come from the central nervous system. Unipolar neurons just have one process that comes from the cell body. Bipolar, two. that come from the cell body processes. Multipolar neurons have got multiple processes that are coming from the cell body. Okay, now this is an axonal terminal. Axonal terminal. In the next slide, I'm going to focus on the axonal terminal because that's where transmission of the nerve impulse comes from. So what happens is that an action potential is going to be processed in the cell body. So this is a cell body. An action potential will be processed in the cell body. So we processed there, and then it will travel along the axon here. So it travels along the axon and it will end up in the axonal terminal. The axonal terminal is the one that relays the action potential across a neuromuscular junction. Okay, so if this here, this is your axonal terminal. Okay, so this is your axonal terminal, and an action potential is coming. So this is an action potential. You know that an action potential is just depolarizing the cell. Okay, so when that action potential is coming along the axon and it reaches the axonal terminal here, what it will do is that it will open the voltage gated calcium channels. So action potential, I'll just write AP, will open voltage gated calcium channels. When it opens the voltage gated calcium channels, calcium is going to enter the axonal terminal from the outside. So calcium is going to enter there. When calcium enters here, what calcium will do is that it will cause a vesicle that is containing a neurotransmitter that is called acetylcholine. So this vesicle that I just drew here, this one contains acetylcholine. Acetylcholine. Okay. So the calcium that enters here will cause this vesicle that is containing acetylcholine to fuse with the membrane of the axonal terminal. So when the vesicle fuses with the membrane of the acetylcholine, it will read the vesicle. So when the vesicle fuses with the membrane of the axonal terminal, it will cause the release of acetylcholine in the synaptic cleft. So now the synaptic cleft is this space. So what I'm drawing here is the membrane for the muscle cell. So this space here between this axonal terminal, the preganglionic axon, this axonal terminal, and the muscle membrane here, this space is what we call the synaptic cleft. Synaptic cleft. So acetylcholine is going to be released in the synaptic cleft. When acetylcholine is released in the synaptic cleft here, what it will do is that it will bind on the nicotinic receptors that are found on the surface of the muscle. Okay, so the muscle end plate here, we call it the muscle end plate. The muscle end plate has got receptors that are called nicotinic receptors. This acetylcholine will bind on these nicotinic receptors. When the acetylcholine binds the nicotinic receptors, what will happen is that ligand-gated sodium channels are going to open. So the sodium channels are going to open, and sodium is going to enter the muscle cell. So sodium will enter the muscle cell here. When the sodium enters the muscle cell, when enough sodium enters the muscle cell, this muscle membrane here that is called the sarcolemma is going to depolarize, meaning that the resting membrane potential of the muscle cell is going to increase. It will shift from -90 to about -70, creating what we call an end plate potential. End plate potential. When this end plate potential reaches, the threshold potential, then an action potential is going to be propagated in the muscle cell. So that is how transmission of a nerve is conducted across a neuromuscular junction. So a neuromuscular junction is just a junction between the nerve here which forms the neuron, between the neuron and the muscle. So neuromuscular junction. All right. So an action potential that is coming down the neuron here, when it reaches the axonal terminal, it will open voltage gated calcium channels, which will cause calcium to enter the neuron. When calcium enters the neuron, this calcium will cause the fusion of this vesicle that is containing acetylcholine with the membrane of the axonal terminal, and it will result in the release of acetylcholine in the synaptic cleft. This acetylcholine that is released in the synaptic cleft will bind to nicotinic receptors. Okay, so these are nicotinic receptors. The nicotinic receptors will then cause a configurational change in the ligand-gated sodium channels, which will cause them to be open. And when the ligand-gated sodium channels on the muscle end plate open, sodium will enter the muscle cell, causing an end plate potential. When an end plate potential reaches the threshold potential, an action potential will be propagated in the muscle cell. And that action potential will now cause contraction, which we're going to talk about in the next video that we're going to do. All right. Now, how is acetylcholine formed? Okay. How is acetylcholine formed so that it can be stored in this vesicle? So acetylcholine, let us draw it from here. Acetylcholine is formed when choline reacts with what we call acetyl coenzyme A. Acetyl coenzyme A. And this reaction is characterized by an acetyltransferase. So what the acetyltransferase will do is that it will get the acetyl from acetyl coenzyme A and to join it to acetyl. So this will result in acetylcholine. This acetylcholine will then be stored in the vesicle. And this is the acetylcholine that is released in the synaptic cleft. Okay, now there are several medical conditions which will affect the contraction of the muscle by destroying nicotinic receptors. So there is an autoimmune condition that is called myasthenia gravis. So myasthenia gravis. In myasthenia gravis, what happens is that the nicotinic receptors are destroyed by autoantibodies. So your body immune system will destroy the nicotinic receptors. And when acetylcholine is released from the nerve terminal, it won't have where to bind. Hence, your contraction of your muscles is going to be very weak, or you'll be easily tired. Another point that you should know is that when acetylcholine is released into the synaptic cleft, after it binds to the nicotinic receptors, there's an enzyme that will come and break down acetylcholine to acetate and choline. That is called acetylcholinesterase. So acetylcholinesterase. So acetylcholinesterase is simply an enzyme that will break down acetylcholine to choline and acetate after it has achieved its function at the neuromuscular junction. Okay, it's the neuromuscular junction. Now, to treat myasthenia gravis, you need to inhibit the acetylcholinesterase so that you can increase the amount of acetylcholine in the synaptic cleft to cause a more sustained contraction. So acetylcholinesterase inhibitors are used in myasthenia gravis so that you can take care of the problem of muscle contraction. Now, there are times where the acetylcholinesterase is inhibited or it is deficient. What that means is that the acetylcholine that was released in the synaptic cleft won't be destroyed. And if that doesn't happen, it means that there will be continuous stimulation of your muscle cell, because your acetylcholine will continuously bind to the nicotinic receptors. So that will cause a tetanic contraction. Okay, there will be tonic contraction and your muscles won't be able to relax because acetylcholine is continuously stimulating your muscles. Okay, so that is how synaptic transmission occurs at the neuromuscular junction. In the next video, we're going to talk about excitation contraction coupling in skeletal muscle and in smooth muscle. See you in the next video.